Minimize. Roland. Obvious.
Hello everyone, good evening and welcome to your own channel Knowledge Hub by Sri Chaitanya, where you are going to get the live, accurate, detailed discussion of the NEET 2024 exam which you have just written. Okay, so stay tuned with us till the last to get the detailed explanation of all the questions, not one or two but the entire 200 questions which you have attempted today in your paper, and follow us till the end. Okay, so we are going to go subject wise with the different subjects, and you are going to get the detailed proper explanation of all the different questions. Here I am Pratyusha, senior botany faculty, and also we have with us.
Hello everyone, very good evening and a very warm welcome to all the students who have joined live with us. I am Dr. Deepak, senior zoology faculty here at Knowledge Hub. Along with me and Pratyusha ma'am, we also have senior expert physics and chemistry faculties, and we all have gathered here to help you with the detailed, authentic and accurate analysis of your NEET 2024 question. So guys, are you all ready for this? If yes, please do let us know in the comment section below. We are waiting for you all.
Okay, so please do hit the comment sections. We want as many as comments for you all so that we can get to know that you are there with us. Okay, so let's make it a bit more interactive. Okay, let me ask you the first question. How was the NEET 2024 paper? I mean, all those who have written the exam last year, according to them, how was the difficulty level? Was it a bit more difficult, or was it like less or moderate? So please hit the comment section and let us know how was your experience of writing the NEET 2024, and also please, all those who have already given the exam last year or that year and getting a comparative analysis from you all, but also those who have just given it this year, for them also, the freshly passed out students from 12th, please let us know how was the paper. How was it writing the NEET 2024 for the first time? How was it giving the NEET exam? Okay.
So all right ma'am, I have a question for you. So last year we had around 20 lakh students who have appeared for NEET examination. And this year the number got tremendously increased and around 24 lakh registrations were there. So, ma'am, how do you think the competition is going to be for this year?
Okay, so every year we can get the analysis that the competition is increasing like crazy. But my dear students, you need not worry at all, because along with the competitors which you are having, your chances of getting selected is also increasing, because we have this year around 1,20,000 government medical college seats. Along with that, the number of AIIMS and other government very prestigious government medical institutes have also increased. They are increasing every year. So it's not very difficult to crack the NEET exam. Just a bit of hard work is required and you can easily do the same. Okay? Yeah.
So ma'am, students might have just come from their exam centers and they must be, eagerly waiting to check their answers and to get their tentative scores. So ma'am, are we going to release the answer key, or how is it going to be?
Yes. So please, dear students, make a note of it along with discussion of the questions and all the different types of questions in all the sections. You are going to find the official answer key right here as an attachment, and we are going to release from our channel, that is Knowledge Hub by Sri Chaitanya. So we are going to publish our official answer key. Please download it and check with your paper which you have given today. So just check with us and you will get the answers of all the questions. So sir will be going to explain you what we are, what Knowledge Hub by Sri Chaitanya is.
So Knowledge Hub by Sri Chaitanya is present in almost all the states across South India. We are currently operating 13 offline centers in all the major cities including Hyderabad, Bangalore, Belgaum, Chennai, Nanded and Vizag. So basically, we offer test preparation services for all the national level competitive exams like JEE and NEET, along with preparing the students of class 8 to 10 for various Olympiad and scholarship exams through classroom, digital and distance learning programs. For more information, please do dial our toll-free number so that you know you are going to get more information.
Definitely. So, we have just introduced another very good program in our knowledge hub by Sri Chaitanya, that is the HyFlex learning program. So, it's not just like any other online program which you might be getting from other different centers. It's like HyFlex means it's a combination of online and offline learning. What you are going to have is that the HyFlex, in the HyFlex courses, you are going to attend the classroom programs from the comfort of your home. You are going to get proper online classes, and that's not going to be like any other. You are going to get a classroom-like experience for all the online classes, and also writing the exams, writing the tests and doubt clarification. You can visit any of our nearby centers. This is particularly for those students who are having a bit distant location, who are actually staying a bit far away from our coaching centers. For them who are not getting to travel every day and to come to our coaching centers, this HyFlex learning program is going to be really a boon. Okay? So, next thing sir is going to tell a bit more about few questions about how was the level and all this.
It really sounds great knowing about HyFlex program, and I feel it's going to be a great advantage, for the students who cannot really come to the centers, maybe because of the distance constraint or maybe because of several other concerns. So before going ahead, ma'am, have you just seen your, basically being a botany faculty, have you seen the botany part of today's NEET paper?
Yeah, I find according to my experience, which we have seen the paper and we have solved also, the paper was moderate. Okay, there were many direct questions from NCERT according to me, but few questions were from the periphery. So we are going to discuss all of them. Okay, students, before that, just one more question for you all. Those who have given the paper this year, how many of you found which part of the paper do you feel is easiest and which was the most difficult? Amongst botany, zoology, physics, chemistry, please hit the comment section fast and let us know. Which part did you find very easy, or directly, which was like directly from NCERT, you did not even need to think twice before attempting such questions, and which was a bit more difficult, and which part did you find very difficult and like very easy? Please just hit the comment sections and let us know. We are also waiting eagerly to know from you all, right?
So ma'am, from student point of view, do you think the paper, the botany part especially, was it easy, was it difficult, or was it moderate?
Yeah, it was a moderate. Like what I have just said, that many questions were directly from NCERT. Along with that, as we are going to discuss more of the questions, so we'll be taking you through each and every question. So let us begin with the discussion of the first question for NEET 2024. So, sir will be taking a leave right now, but he is going to be with us, so don't worry, he will come back for the zoology part. So, sir?
Yeah. So, students, see you all in the zoology section. So, I'll be discussing the zoology part. Until then, bye-bye.
Yeah. So, let us begin the paper discussion and with the analysis of the NEET 2024 paper. So, let us start with the botany section first. Okay, so here we are with the very first question which you all have. Okay, before that, let me just select. Okay, so first question. This is directly from the biomolecules chapter. So the question says that inhibition of succinic dehydrogenase enzyme by malonate. This is exactly the line from the NCERT, which is a classic example of what given under competitive inhibition. And so you know this is a competitive, there are different types of enzyme inhibition, competitive, non-competitive and uncompetitive type. Out of them, this is going to be the competitive inhibition, which is directly an example which is given from. So this is a straight away question from NCERT. And the option is option number 1. We are eliminating option number 2 and 3 because it's not cofactor inhibition, there is nothing like that, and also it is not a feedback inhibition. Let's go to the next question. I hope this question was very, very clear for you all, right? Okay, so let's go to the next question. So, I hope the board is visible to you all. Which one of the following can be explained on the basis of Mendel's law of dominance? So, exclusively the catch here is that we are talking about law of dominance and not any other law. So, law of dominance definition you can refer to NCERT. What is the law of dominance? What is the definition of law of dominance? Now, let us see what are the options. One out of one pair of the factors, one is dominant and the other is recessive. Then the next one is alleles do not show any expression and both the characters appear as such in the F2 generation. Okay, so here we can see that we have to be taking the factors which is depending on the basis of law of dominance. Next thing is factors occur in pairs in the normal diploid plants. So this is correct. This is also correct. This is not correct because alleles do not show any expression and both characters. So both characters cannot appear. Right. Next thing is the discrete unit controlling a particular character is called a factor, which is also true. Mendel had named them as factors before they were called as genes. Okay, so this is correct. And the next thing is the expression of only one of the parental characters is found in a monohybrid cross. So that is also correct according to the NCERT. So this is, you can also say, a direct question from NCERT and nothing is in the periphery, it's from NCERT directly. So A, C, D and E is correct. So the correct answer option here is option number 4, which is giving A, C, D and E. Okay, let's go to the next question. I hope the explanation is very, very clear. And let us go to the next question. So here we feel that lecithin, okay, this is also a question straight away from the biomolecules. Lecithin, a small molecular weight organic compound, is found in the living tissues. So in the biomolecules, you must have heard about lecithin, which is an example of what? It is not going to be an amino acid, it is not glycerides, it is not carbohydrates either. It is definitely a phospholipid, right, my dear students? So let us go to the next question of here. So question number this, so which we are going to follow, match the list 1 with the list 2. Let's see what we have in list 1 and correspondingly which we are going to have in the list 2. So list 1 we have nucleolus. Okay, so nucleolus, we know that this is the site for active ribosomal RNA synthesis, right? Next thing is we have centriole. Centriole is what? So if you see the organization of centriole, it is organized in the form of a cartwheel-like appearance, right? Then after that we have leucoplasts. So about leucoplasts, we can say that this is required in some of the, like there, for storing nutrients. Okay, so leucoplasts goes with the storing of storage. We have a lot of leucoplasts in potatoes and various such things. So for storing nutrients, leucoplasts is going to be there. Next one is Golgi apparatus. So Golgi apparatus, we also call it a packaging organelle. This is going with the last remaining option, that is the site of formation of glycolipid. Hence here the correct match is option number 3, which we are going with A3, B2, C4 and B1. I hope I am very, very clear, right? Let's go to the next question. Here we have which of the following is an example of actinomorphic flowers. So you have seen all the family and you have also seen the different flowers coming from the family. We have flowers which belong to actinomorphic, which is coming under zygomorphic, or which can be divided into 2 parts from the center, and actinomorphic means it is radially symmetric, right? And also asymmetric flowers are there. Out of these options, there is only one flower which is actinomorphic or which is radially symmetric, that is Datura. rest of the flowers, whether it is Pisum or whether it is Sesbania or Acacia, all of them are zygomorphic flowers, right? So this was also a very simple direct question from NCERT, right? So far, how are you finding the botany paper? So far it is very easy, right? Easy, moderately easy, slightly easy, or a bit difficult. Please let us know in the comment sections, right? So that we can give a better explanation, right? According to that, we will proceed. Okay, so let's come to the next question. Here, 106 we have, how many molecules of ATP and NADPH are required for every carbon dioxide molecule which are fixed in the Kelvin cycle? Okay, so in the Kelvin cycle, how many are we having? Whether we are having, how many molecules of ATP are required and how many molecules of NADPH? Okay, so we know, again a direct question from NCERT, by the calculation base we can get that we require 3 molecules of ATP and 2 molecules of NADPH, which is required for every carbon dioxide. Just for every carbon dioxide which is fixed in the Kelvin cycle, we have this, right? Okay, my dear students, let's go to the next question explanation, right? So far so good. Any questions? Please hit the comment sections again, right? Okay, so formation of interfascicular cambium. Now, interfascicular cambium is a type of cambium. Cambium, how it is formed? Everyone, this is a very common question. It's just coming from the anatomy of flowering plants. So, interfascicular cambium from fully developed parenchyma cells. Parenchyma cells are what? Permanent tissues. And interfascicular cambium, it's a type of meristematic tissue. So, how it is happening? This is an example of what? So, we know that from permanent, okay, if we are going to back to meristematic, so what happens here? This process is known as dedifferentiation, whereas the other process, maturation, differentiation and redifferentiation, everything is a formation of meristematic to permanent, right? Very clear? Okay, thank you students. So let's go to the next question here. So auxin is what? This is a type of plant growth regulator. So this is coming from the plant growth and development chapter, right? So auxin, this plant hormone, this is used by the gardeners. Very commonly we are using auxins by the gardeners. These are used to prepare weed-free lawns. So now, my dear students, this is a tricky question according to me. Okay, so if we are going to prepare a weed-free lawn, okay, but no damage should be caused to the grass. So grass is what? A monopod plant. And we want to eliminate the weeds, right, from the lawns. But the grasses should be kept proper. There should be no damage done to the grasses. So what property of the auxin shall be utilized? Okay, now here we have different types of auxin growth properties. Number one, it does not affect mature monocotyledonous plants, which is true. Right, we are keeping it aside. Let's see the other options. We have, it can help in the cell cycle division in the grasses to produce growth. Here we are not looking at the growth, but we are looking at specially a weed-free lawn. Okay. Next thing is it promotes apical dominance. This is also true for the auxin, but we are particularly looking for this property, not for any other property, my dear students. So another one, promotes abscission of mature leaves. So now this is not a characteristic of the auxin. This is done by some other plant growth regulator. Now, so which one do you select? Here, based on the property which we are looking here, this is going with the option number 1. Yes. So here it does not affect the mature monocotyledonous plants, which is the grass. Remember, grass should not be affected. At the same time, it should remove the weed. Okay. So here the answer is option number 1. Let's go with the next question here. So which of the following are required for the dark reaction of photosynthesis? So dark reaction, we know this is a type of light independent reaction. I mean, we are dependent on the products of the light reaction, but we are not directly dependent on light or any other things which can trigger the formation of the compounds which is related to the light reaction. So what we are looking for here, right? So light definitely will not come in any of the options, right? And also chlorophyll, exclusively it is present in the thylakoid lumen of the grana and it does not occur in the stroma. Hence chlorophyll also is eliminated. Rest we are left with what? Carbon dioxide, ATP and NADPH. Definitely we require this for the dark reaction of photosynthesis. Hence we are left with this answer, that is answer number 1. In this case, which is C, D and E only, right? Okay. So far I hope you have got many answers to all your queries, right? Let's go to the next question here. So any confusion here? Any other answers, right? No? Shall we proceed further? Yes, good to go? Okay, let's go. So the next thing here, now this is a question from the molecular basis of inheritance lactoperon concept, which must be one of your very favorite concepts, right? Yes. So the question is, the lactose present in the growth medium of the bacteria is transported to the cell by what? By the action of what? By the action of, so we have 3 different types of the enzymes which are helpful for the intake of lactose. Amongst them, permease. This helps in the transportation of the lactose inside, right? Whereas polymerase is not linked with lactoperon. We don't have beta galactosidase is doing some other property. Okay, it's breaking lactose into glucose galactose, that thing, but it is not taking it, it is not transporting it inside. Okay, and acetylase is also, it's not related to the transport. Hence, the answer is number 1. Okay, let's go to the next question. So, last question was also a very direct question. I hope you enjoyed this one. Okay, the cofactor of the enzyme carboxypeptidase. What is the cofactor here? So, here this is not a direct question. This is a bit from the periphery. But if you have solved and read NCERT along with that few test papers, last year papers and all of this, you could have got this question very well. So the answer is, I am directly giving you the answer because there is no other third option or second option. This is zinc, which is the cofactor of the carboxypeptidase enzyme, right? So for this you can go quickly and just see the enzymes chapter of biomolecules. So quite a few questions are from biomolecules this year. In the botany section, we have got a good weightage of the biomolecules, right? Let's see the next question. This is also one of the very easy questions which I feel that here you have to identify the part of the seed in the given figure which is destined to form the root. Okay, root when the seed is germinating. Here they have labeled 4 parts. One of them is going to be, so we have A, B, C and D. So B is standing for the plumule and A is the coleoptile. C is the radical whereas D is the coleorhiza. Right, so which is developing into the seed? This is from the seed germination. You have also read it in your lower classes. Not only in this 11, in the morphology but also in the lower classes. This is going to be option number 1, that is C, which is actually the radical. So remember my dear students, radical is the only part which is destined to form root when the seed is germinating, right? Okay, so any doubt? I hope you will not have any doubt in this question. Even those who have not, even if those who have done a bit least preparation can also answer this question very well, right? So this was tremendously easy. Okay, let's see the next question here in front of you. So given below are 2 statements. So this is a statement type question. Once again, so statement type question, let me read the question first, let me read the statements first and then we will see the options. Okay. So statement number 1, it is chromosomes become gradually visible under light microscope. So chromosomes, if they are gradually getting visible under light microscope during the left protein stage. In left protein stage, what is happening? So left protein, after that, the next statement is that the beginning of the diploidine stage, this is recognized by what? By dissolution of the synaptonymal complex. Okay, so you can see, yes, it is true, the chromosomes are gradually becoming visible under the light microscope during the left protein stage. Yes, that is true, it becomes visible during the left protein stage only, right? And then beginning of this is dissolution of synaptonymal complex. Okay, so now we have to choose the correct options from the statements. 1 is true, 2 is false. Next option, 1 false, 2 is true. Third option is correct, which is both the statement 1 and statement 2 are true. Okay, so the option number 3 is correct. Here we are going with statement 1 and statement 2. Both are true. Right, my dear students? This is also a very direct NCERT question. Okay, so just you can see, if you have read the NCERT very well, you could have solved so many questions so far in the botany section. Right? Okay. So let's see the next question here. So for this, now this is a question from the biodiversity. Okay, so the type of conservation in which the threatened species, these are taken from the natural habitat and taken out. Okay, taken out from the natural habitat and they are placed in the special setting where they can be protected. Okay, and they are given special care. So this is a bit tricky. Here, what is the catch? They are taken out from the natural habitat. Okay, so if they are taken out from the natural habitat, it cannot be by any chance in situ. In situ means they are protected in the natural habitat only. Right. So if they are taken out from the natural habitat, so we first of all we are going with the elimination. We are deleting this option. Okay. Next, what we are left with, semi-conservative method. It doesn't come under this kind of type. And the next thing is sustainable development. This is also not true for this. So finally we are left with this one. This is called the biodiversity conservation. So the correct answer is the option number 4. Okay. So, I hope you all got this solution. Was it very clear? Right? Okay, let's move to the next question. So, now this is again a statement type question. And the question here is, now see, you are having 2 statements, right? The first statement is that parenchyma, this is living, but collenchyma is dead. Is it true, my dear students? We all know which are the living and which are the dead tissues. So, definitely if we just look at the first statement, we can get that parenchyma is also living and collenchyma is also living. Only sclerenchyma is dead. Right. Now the next statement is gymnosperms. They lack the xylem vessels, but presence of the xylem vessels is the characteristic of the angiosperms. Okay, so gymnosperms, yes, they do lack the xylem vessels. We know that they don't have the xylem vessels as such, but angiosperms is having the xylem vessels present in them definitely. Okay, so xylem vessels is not present in gymnosperms and it is present in the angiosperms. So we can see from here, this statement is not true because collenchyma is a living tissue. Statement 1 is false and statement 2 is true. So we are going with option number 2 here. Right? Okay. Let's go to the next question. So here, this is a question from this, like one of the ecologies, and we can get this question. This is directly and indirectly NCERT question. Okay, there is nothing to solve. You just need to tell us what in this equation is K. Okay, K is coming here 2 times. This is Rn is K minus N by K. Okay, so K is what here? You don't need to solve any equation, you just need to know what is K, which I think all of you already know. Direct NCERT question again. K is nothing but the carrying capacity, right? So let's move forward to the next question. Here we have question number 117. Let's see, let's go match list 1 with that of list 2. Okay, so we have a name of different types of organisms and we have the list of them. So amongst them, Clostridium butylicum, this is responsible for generating butyric acid. Next thing is Saccharomyces cerevisiae, this is utilized for ethanol or for alcohol production in large scale in the industries. Next one is that Trichoderma polysporum, this one is responsible for producing cyclosporin A, and the next one Streptococcus species, this generates the clot buster, that is streptokinase. Okay, so now let's see, this one is going with option number 1. Yes, so you all also just give the answers directly so that we can just give on the interaction section, right? So far so good, people. Let's move to the next question. You have got quite a few as the following statement types and all these questions, which might be very interesting for you all. Okay, let's see this question. This is which of the following is not, okay, this is not a criterion for classification of fungi. Now, my dear students, you might be just missing out these small things in just a bit of hurry. Okay, so here the catch was not. It's not a criterion for classification of fungi. Definitely we have mode of spore formation. This is a classic characteristic. Next one is fruiting body. It is also same, we know fruiting bodies, depending on that fungi is grouped under different groups, and also morphology of mycelium, whether septate or aseptate, you are having different groups of the fungi. So we are left with mode of nutrition. This is not a criterion for the classification of fungi, right? Okay, let's see the next question here. So here it is, the capacity to generate a whole plant from any cell of the plant, it is called what? If we are generating a whole plant from the any type of cell of the plant, this is called as totipotency, the term which is given directly in NCERT. It's not differentiation, it's a bit different. As I have already explained in one previous question, differentiation means from permanent cells you are getting back the meristematic cells, and also it is not somatic hybridization. It is also not the micropropagation. Okay, so we are going with the third option, totipotency. And if anyone feels anything else which needs to be added and all, you can always hit the comment section. Yes? Okay. So let's move to the next question. So far, see. Here is the next, again, the statement type question. This is from biotechnology chapters. Okay, here we have Bt toxins. These are a group, these are insects group specific, right? We know Bt toxin released by Bacillus thuringiensis. These are the insect group specific, definitely, and they are coded by what? The gene crygene, IAC. Okay, next statement says that Bt toxin, it exists as a inactive prototoxin in the Bacillus thuringiensis. Hence, the bacteria is not affected, right? We have studied this. However, after ingestion, okay, after ingestion, what happens? By the insect, the inactive prototoxin gets converted into the active form due to the acidic pH in the insect gut. Now, where is the catch here? This is, okay, so this is not due to the acidic pH in the insect gut, but this is due to the alkaline pH present in the insect gut. Hence, statement number 1, you can see this is absolutely true. There is no doubt in this. But statement number 2, we cannot consider this to be true. Hence, the answer is number 1, statement 1 is true, but statement 2 is false. Right? Okay. So let's move forward to the next question. Here we have, this is identify the set of correct statements. You have to identify the set of correct statements out of this, okay, out of the few statements. Okay, so we have this, the flowers of Vallisneria, they are colorful and they produce nectar. Is it true, my dear students? Okay, let's see the options. Next one, the flowers of water lily are not pollinated by water. Okay, next one, in most of the water pollinating species, the pollen grains are protected from wetting. Okay, so we can see here that, see, flowers of Vallisneria need not produce colorful, need not be colorful and produce nectar because they are not pollinated by any insect or any other biotic agents, right? So they can go without being colorful and produce nectar. So this is not a correct option. The other one, water lily, this is not pollinated by water, but it is pollinated by insects. Okay, we know this. Next thing is that, so most of the water pollinated species, the pollen grains are protected from the wetting, which is an important characteristic of the water pollinated species, right? So it has to be protected from wetting. Then the next one is that pollen grains of some hydrophytes are long and ribbon-like, which is also true, right? So this is one, and we have in some hydrophytes the pollen grains are carried passively inside water. Okay, so inside water we have that most of the water pollinated species. Okay, wait, let's see the correct options from here. This is not, we are getting the set of the correct statements, right? So this statement is correct, saying that the water pollinated species pollen grains are protected from wetting. This is true. Let's see the next set of correct statements. Pollen grains of some hydrophytes are long and ribbon-like, which is also true. And in some hydrophytes, the pollen grains are carried passively inside water, which is also true. But these two options are incorrect. Okay, so we are getting the correct answers from the following. Sorry, a bit change of here. So I have actually explained you that option number B is also correct, which is water lily, not pollinated by water, insect pollinated, and this is prevented from wetting. This is correct. This is also correct, and E. So we are going with the option number 2, which is having B, C, D and E as the correct set of options. Okay, now let's move to the next question. So a transcription unit in DNA. Okay, this is defined primarily by the 3 regions. So we know if it is a transcription unit, it will be having 3 regions. First, it will be having, represented like this. This is going to be a promoter, okay, followed by the structural gene which is getting transcribed, and this is followed by the terminator. We can straight away look in the answers which one is having promoter, structural gene and terminator. This is option number 2. Okay, the other ones, it's not directly related to the transcription. Only in a transcription unit will be having these 3 answers. Okay, fine. Now let's go with the next question. So bulliform cells, these are what present in the monocots, we know, and this is present in the monocot leaf. If you see the anatomy of monocot leaves, you will find the bulliform cells, right? And they are responsible for increased photosynthesis in the monocots and providing large spaces for storage, inward curling of the leaves of the monocots and protecting the plants from the salt stress. So direct question, we can also see that bulliform cells, this is responsible for the inward curling of the leaves in the monocots and not it is going with any other property, right? So I hope I am very clear. Let's go with the next question. Okay, so next question is identify the types of the flower based on the position of the calyx, corolla and the androecium also, okay, with respect to the ovary. So we are actually going with the ovary position and any other type of floral holes. You can see here in the diagram, this is just directly a diagram based question from NCERT. If you look at the different types of the diagrams, which is epigynous, perigynous and hypogynous, this one, the level of the ovary is parallel. Let me change the color once so that it's a bit more visible. See, the ovary is here. Okay, this is going parallel with this, right, with one of the floral holes, that is the androecium, and it is also coming parallel with the next floral hole, that is the corolla, and also the calyx. Also in this we are seeing the ovary, this is coming parallel with the 3 floral holes. So in both the cases, this is perigynous, perigynous. This is also denoting perigynous condition and this is also denoting perigynous condition. Okay, so the correct answer is option number 2, that is perigynous. Okay, I hope I am clear. Let's go with the next question. So here, the fate of a piece of DNA carrying only the gene of interest. Okay, so the piece of DNA which is carrying the gene of interest, which is transferred into an alien organization. This is a biotechnology question. This one is what? So we have to eliminate, we have to go with the correct answers from the options given below. Okay, so we can see the piece of DNA would be able to multiply itself independently. This doesn't match. Okay, but this is correct that it may get integrated into the genome of the recipient. It is correct. And also it may multiply and be inherited along with the host DNA. Okay, which is also true. Let's see the other option. The alien piece of DNA is not an integral part of the chromosome. This is not possible for the recombinant DNA technology. And also the last thing, it shows an ability to replicate. It's not self-replication, it is along with the genome of the recipient, it's going to replicate. Okay, so amongst this, B is correct, C is correct, so we are choosing the correct options. Hence, we are going with option number 1, which is having B and C only. Rest of them are not the correct answers. Okay, so my dear students, I hope you have really enjoyed this, and let's come with the next part of botany, which will be explained by Vaishnavi ma'am. Okay, so I am taking a leave from you all, so good luck with the questions.
Thank you, Pratyusha ma'am. So, hello everyone, this is Vaishnavi, Senior Botany Faculty from Knowledge Hub by Sri Chaitanya. So, let us move on to the next questions. We'll discuss the next questions over here. Question number 126. The tropical regions show greatest level of species richness because. So, we know that in the tropics, the species richness is very high. Why is the species richness very high? They have given here some options. We need to choose the reason. So, A. The tropical latitudes have remained relatively undisturbed for millions of years. Hence, it has given more time for the species diversification, more time for speciation. So, the statement A is right. Now, we'll look into the second statement. Here is the second statement. Tropical environments are more seasonal? No. Tropical environments, they are less seasonal. So here this more seasonal is given, so the statement B is wrong. We'll look at the C statement here. Okay. So in the C statement, what did they give? More solar energy is available in the tropics. That is right. So sun rays, so the solar energy will be directly falling over the tropics. So that is also one of the reasons for being more rich in terms of species diversity. So C is right. Then D. Constant environment promote niche specialization. Yes. So D is also right. Then coming to the E statement. Tropical environments are constant and predictable. Yes. That is also one of the reasons for why tropics are more rich in terms of species diversity. So what all are right here? A, C, D and E. A, C, D and E means it is option 3. I hope it is right for you all. Now we'll move to the next question. Question number 127. Observe this question. In this question, it is talking about the flower color of snapdragon. So the flower color of snapdragon is coming under the topic of incomplete dominance from Principles of Inheritance and Variations chapter. So we'll look into the question now. The question is saying pink color snapdragon. Okay, pink color snapdragon means capital R, small r. This is the pink color snapdragon. Was crossed with a red flower snapdragon. So red flower snapdragon will be capital R. This is red color snapdragon. The pink color snapdragon will produce two types of gametes. So what are those two types of gametes? One gamete carries capital R, other gamete carries small r. Red parent, it produces only one type of gamete and that gamete is carrying capital R. Let us fuse the gametes now. If we are fusing the gametes, capital R gamete of pink parent fuses with the capital R gamete of red parent, we get the offspring genotype as capital R, and capital R, we already know that it is red. Now, other gametic fusion we will see. Smaller gamete of pink parent fuses with the capital R gamete of red parent, and what do we get the genotype of the offspring here? We are getting capital R, small r, and it is going to show pink. Here, when two dissimilar alleles are there together, what is being expressed? Neither capital R nor small r, it is showing pink color, neither red nor white, pink, and this is due to incomplete dominance. So in the product, what are we getting then? We are getting red and as well as pink color flowers. So that means red as well as pink. So that means option 4 is the right answer. I hope you all are getting this. Now we'll move to the next question. Question number 128. Look at this question number 128. Major causes of biodiversity loss. Major cause of biodiversity loss. Let us see. Overexploitation. Yes. We know that we are dependent on the organisms. When our need turns into greed, then only the overexploitation is happening, and that is one of the reasons for the loss of biodiversity. Let us look into the next option, B, co-extinction. So the organisms which are in obligate relationship, where one is definitely dependent on the other and it cannot live independently. Such mutually dependent organisms, obligate relationship having organisms, in them, if one organism is getting eliminated and then parallelly the other organism who is dependent on them also gets eliminated. They are eliminated together. That means co-extinction. Yes, this is one of the reasons for the loss of biodiversity. Mutation? No. Habitat loss and fragmentation? Yes. So it is because of human colonization only. We are constructing the roads, buildings and all, because of which the habitat of the wildlife is being fragmented. The habitat of the wildlife is being lost. This is considered as one of the major loss of biodiversity. There are totally 4 losses, 4 reasons are there for the loss. This is one of the major loss. Important loss under the evil quadrant. Now coming to migration, no. So what are there here? A, B and D. Or that means option 2. I hope you have answered this right. Now we'll move to the next question. Question number 129. Okay, this is the match the following question and it has come from fungi, that is from the biological classification chapter. We'll see them. Let's try to match it. Rhizopus. Rhizopus, this is belonging to Phycomycetes, Zygomycetes. That means Rhizopus is matching with the 3 in list 2. It is called as bread mound, belonging to Phycomycetes. Eustilago. Eustilago is belonging to the Basidiomycetes member, and Eustilago is also called as rust fungus. So B is matching with 4. Puccinia. Even Puccinia is belonging to Basidiomycetes. Puccinia is called as smart fungus. Basidiomycetes member only. Fourth option, Agaricus. Agaricus bisporus. It is a mushroom, which is also belong to Basidiomycetes only. So what is the right option here? A is 3, okay. Then B is 4. B is 4. I think here the option it should be B4, and C is, it is 2, and D is 1. So option 3 is correct over here. Now we'll move to the next question, guys. Question number 30, and here it is again a question from principles of inheritance and variations. Look at this question. Black seed color is dominant over white seed. They are also mentioning that black is dominant over white seed. Black seed is expressed both in homozygous dominant and also in heterozygous. So dominant over white seed. In order to find out the genotype of the black seed, now black seed color is visible but its genotype is unknown. It can be either capital B or capital B small b. What is there inside the seed? We don't know. So in order to know the genotype of the unknown, we will be having to do the test cross here. In the test cross, the cross should be done with the recessive parent. Here the recessive parent is white color seed. Okay. So white color seed means small b, small b. So this is the right answer. Now we'll move to the next question, ma'am. 131. Look at this question. 131. It is from anatomy. Look at this question, diagram based question. So far we got one diagram based question, and this is from anatomy. Let us try to label them and then we'll look into the answers. This A is nothing but epidermis. Look at B. B is subsidiary cells, or otherwise called as axillary cells also. Look at the C. C is the God cells. And D. D is what? It is showing the stomatal pore, or otherwise called as stomatal aperture also. Now we'll look at the question. In the given figure, which component has thin outer walls and thickened inner walls? The differential in the thickening is found in the walls of God cells. In whom the differential thickening is found? It is found in the walls of God cells. Okay. So particularly here they mention that the outer walls are thin and inner walls are thick. That is found in dicot God cells. So God cells means it should be C. Now we'll move to the next question, 132. The list of endangered species was released by. The endangered species, it is released by IUCN, which is the International Union for the Conservation of Nature and Natural Resources. 133. Again we are going to have a match the following kind of question. Okay. So in the match the following kind of question, the first one. 2 or more alternative forms of a gene is called as allele. So A is 3. So A is what, ma? 3. Next B. Cross of F1 progeny. Cross of F1 progeny with the homozygous recessive parent. With the homozygous recessive parent is test cross, so B is 4. Then we'll look at C. The cross of F1 with any of the parent is called as back cross. Then D. The number of chromosome sets in a plant It is ploidy level, whether one set is there, two sets means diploid, three sets means diploid. Likewise, the sets of the chromosomes, if they are asking, it is related to ploidy. So what is here? A3, B4, C1 and D2. That means the answer is option 1. A3, B4, C1 and D2. I hope you all have answered it very well. Now I'll be moving to the next question. That is 134. So in this 134th question, hint 2 always cuts the DNA molecule at a particular point called recognition sequence, and it consists of, now this is fact-based question, so how many base pair sequence are present here? That is 6 base pairs. It is a fact-based question, so there is no second thought on this question. Now moving on to the next one. Here is the next one, 135th question. 135th question, let us look into that. Spindle fibers attach to the kinetochores of the chromosome during. This is the question from the cell cycle and cell division, mitosis, the chromosomes. Spindles are attaching to the chromosomes. Which part of the chromosomes now? They are attaching to the kinetochore. And this is all happening in which phase? In the second phase of mitosis. And what is the second phase of mitosis? It is metaphase. So the answer is option 4. See, such a simple question it is. I hope you all have marked right in this. Now I move on to the next question. 136. See, once again match the following question. Again, it is also from ecology chapter, which is having more weightage. I hope you all know about this. So 136, Robert May. This scientist, Robert May, has given a conservative estimate of how many species are present. He said that there are 7 million species that are present globally. That is the conservative estimate given by the scientist Robert May. Now, moving on to the second one, Alexander von Humboldt. This person, he has given the species area relationship. He said that the more the area he explored, more is the species richness. Once again, he said that more the area he explored, more is the species richness, and this also up to a particular limit, given by Alexander von Humboldt. Then Paul Ehrlich. The scientist Paul Ehrlich has given Rivet-Popper hypothesis. In this Rivet-Popper hypothesis, he tried to compare the ecosystem with the aeroplane in order to prove that species richness or diversity is having so much of importance in the ecosystem. So that Rivet-Popper hypothesis is given by whom? Paul Ehrlich. Next, David Tillman. David Tillman has given a long-term experiment in outdoor plots. It's not a short-term experiment. It is a long-term experiment. It cannot be done in a day or two. It requires a lots and lots of days are required. It's a long-term experiment which is performed in outdoor plots, not inside the laboratory. So, D is 2. So, 3, 8 is 3. Alright. B is 1, C is 4 and D is 2. So, the answer would be option 4. Moving on to the next question here. 137. Once again the match the following question. See, we are finding many match the following questions over here. Okay, let us see. This question is coming from molecular basis of inheritance. Let us see. So, Frederick, he is a scientist. So, this scientist has given the transformation. It is the Griffith. Griffith is a scientist who has given the transformation. He is Frederick Griffith. Frederick Griffith has given the transformation. So, A should be 3. Then B, Jacob and Monod. Jacob and Monod, they proposed lac operon. Alright. Now coming to Hargobind Khurana. So this person, Hargobind Khurana, had a role in the genetic code, in proposing the genetic code. So C is 1 and D. Michelson and Stahl's experiment. The scientist has given the semi-conservative model of DNA replication. The semi-conservative model of DNA replication in E. coli using heavy isotope of nitrogen given by Michelson and Stahl. So it should be A is 3, then B is 4, C is 1 and D is 2. The answer is option 4. Next question 138, once again match the following. Monodelphous stamens. Monodelphous stamens are found in hibiscus, which is belonging to Malvaceae family. So Malvaceae family characteristic feature is having monodelphous stamen. Malvaceae family example given here is China rose, hibiscus, China rose, hibiscus. Diadelphous stamens. Diadelphous stamens is a characteristic feature of Fabaceae family. So Fabaceae family member, we have to check who is there. Tell me, who is there? It is P. So 2. Polyadelphous stamens. Polyadelphous stamens are found in citrus. Polyadelphous means what? Many bundles of stamens if they are formed, it is polyadelphous. Diadelphous, 2 bundles are formed due to the fusion of stamens. Monodelphous, there is only 1 bundle. Epiphyllous. Epiphyllous, it is found in leaf. Epiphyllous or epitepalous both are same, which means that they are having undifferentiated calyx and corolla or undifferentiated sepals or petals. So D is 3. So A is 4, B is 2, C 1, D 3. And the answer is option 3. We'll move to the next question. 139, once again match the following. Look at this list. GLUT4. This GLUT4 is acting like an insulin receptor. Alright? So it is acting like an insulin receptor, which means it enables glucose transport into the cells. Then B. Insulin. Insulin is an hormone which is released by pancreatic beta cells. Trypsin. What is trypsin, ma? Trypsin, it is considered to be as an enzyme which is involved in digestion. It's a digestive enzyme. Then collagen. Collagen is an intercellular ground substance, yes, which is found only in animals. So D is 3. 4, 1, 2, 3, 4, 1, 2, 3, option 3. I hope you all have marked it right. Moving on to the next question. 140. Which of the following statements is correct regarding the replication of E. coli? What do you mean by replication? Replication is a process by which DNA gives rise to another DNA molecule. That is called replication. Now see, in replication, obviously the enzyme that is needed is DNA polymerase only. So look at the options. If you look at the first option, they are saying DNA dependent DNA polymerase, that is right, but not in both 5' to 3' and 3' to 5'. It is done only in 5' to 3', never in 3' to 5'. Let it be replication or transcription. Whatever it is, the polymerization takes place always in 5' to 3' direction only. No matter it is replication or transcription, it is constant. So A, no. First option is not the right answer. Second option, DNA dependent DNA polymerase, right. 5' to 3', right. I just told you in the first option only, so this is the right answer. Let us just look into the third and fourth options as well. In the third option, they are saying 3' to 5'. I already told in the first option that it is wrong. And look at the fourth option, they are saying RNA polymerase. Why is RNA polymerase coming in the picture over here? It is a replication and all we need is DNA polymerase. Nothing has to do with RNA polymerase. 141, see here, one more match the following question. And this question, it's from the topic of respiration, physiology, plant physiology. See, citric acid cycle, also called as tricarboxylic acid cycle. It is also called tricarboxylic acid cycle, and this process is solely taking place in the mitochondrial matrix. So where is the mitochondrial matrix? A is 2. Glycolysis. Glycolysis, glucose converted into 2 molecules of pyruvic acid. And this process is the only process which is taking place in the cytoplasm, and this is the only step which is common between both prokaryotic and as well as eukaryotic respiration. Where it is occurring, I said, it is taking place in the cytoplasm. So where is the cytoplasm? 1, right? Now third one, electron transport system. So where is this electron transport system taking place? It's the last step of respiration, eukaryotism. And this last step is going to happen in the inner mitochondrial membrane. And look at the last one, the proton gradient. This proton gradient is found in the space of intermitochondrial space of mitochondria. Intermitochondrial space is what? The space which is present between the outer and inner membrane of mitochondria. There we can find the gradients. So it is 3, 2, 1, 4, 3, 2, 1, 4, 3, option 4. I hope you all have marked it right. Moving on to the next one. Identify the step in tricarboxylic acid cycle. Just the continuation of the previous question. This is also coming directly from respiration chapter. TCA cycle, where is it taking place? Citric acid cycle or tricarboxylic acid cycle? In the eukaryotes, in the mitochondrial matrix of eukaryotes, which does not involve oxidation. Isocitrate to alpha-ketoglutarate acid, yes, oxidation needed. Malic acid to oxaloacetic acid, yes. Oxidation is taking place with the production of oxaloacetic acid from malic acid. Succinic acid to malic acid, yes, this is also an oxidation step. So what is the odd one out over here now? Option 1. So in this only, oxidation is not taking place. Then what is the step called? Succinyl coenzyme to succinic acid. That step is called as substrate level phosphorylation. In the substrate level phosphorylation, coenzyme is removed. Here GTP is formed. That is what happening in the substrate level phosphorylation. So answer is option 1. 143. Spraying of sugarcane crop with which of the following plant regulators increases the length of stem, thus increasing the yield? So this is the function done by gibberellic acid, gibberellins, gibberellic acid, right? So when is gibberellic acid sprayed on the sugarcane plant? Obviously the stem is what important because it is having the carbohydrates, it is having the sugars. So the length of the sugarcane stem is increased so that the yield of the sugarcane also can be enhanced. So the answer is gibberellic acid. 144. The DNA present in the chloroplast is, it is double-stranded innate circular DNA. Double-stranded innate circular DNA. So, this is the answer, option 4. 145, again this is a statement-based question we are going to see. Two statements are given. Statement 1. In C3 plants, some oxygen binds to rubisco, hence CO2 fixation is decreased. Why is this happening? When rubisco binds the oxygen, you know what is going to happen, right? What happens? Rubisco is a dual enzyme. It has the affinity for both CO2 and O2. It has the affinity for both carbon dioxide as well as oxygen. When it is binding with the carbon dioxide, no problem, nothing. Happily the photosynthesis will take place. All the problem will happen when it is binding to oxygen, because when it is binding to oxygen, it is going to cause photorespiration. And what does the photorespiration do? It is going to decrease the yield of photosynthesis. And why? It is because what is done by the photosynthesis is undone by the photorespiration. In photosynthesis, what is happening? Carbon dioxide is being fixed. And what is happening in the photorespiration? The same carbon dioxide is lost in mitochondria. So, in C3 plants, some oxygen minus rubisco, hence CO2 fixation is decreased. That is right. Look at the next statement. In C2 plants, mesophyll cells show little photorespiration, while bundle sheath cells do not show any photorespiration. No, this is incorrect. Alright. So the answer, statement 1 is right but 2 is false. That means state option 1 is right. So this is all about the botany question paper. I hope it all went well with respect to botany. Oh, I'm so sorry. So we'll move to the next question. This is 145th only, no? 146. Which of the following are fused in the somatic hybridization? Somatic hybridization is going to involve the fusion of protoplasts. So protoplasm is what? It is the cell without cell wall. That is protoplast. Two protoplasts are fused in the somatic hybridization. So the answer is option 1. Moving on to the next one, 147th question. Statement based again. Okay, in the statement based, statement A, asexual reproduction occurs usually by biflagellated zoospores. This statement based question is based on the Phaeophyceae class of alga. This Phaeophyceae class means it is the brown alga. So in brown alga, asexual reproduction happens with the help of biflagellated zoospores. That is right. Two flagella, they are laterally inserted. So one, statement A is right. B, sexual reproduction is oogamous only. No, not just oogamous. The other types of fusions are also possible. As they mention the word only, the statement is going to be incorrect. A is right, B is false. Then C, stored food in the form of carbohydrates, which is either mannitol or laminary. Yes, that is the reserve food material found in brown alga. True? Then D, the major pigments found in whom? Brown alga are chlorophyll A, C and carotenoids like xanthophyll. Yes. These are the main pigments that are found in brown alga, that is Phaeophyceae class of alga. E, vegetative cells have cellulosic wall usually covered outside by the gelatinous coating of algae. Yes, cell wall, internally it is cellulose and externally it is having a phycocolloid, that is algae. So E also is right. Only B is incorrect here. That means option 1. This is 147th question. Now 148. In an ecosystem, if the net primary productivity of first trophic level is 100x kilocalories per meter square per year, what would be the GPP of the third trophic level of the same ecosystem? Here this is the question based on 10% law, also called as the Lindemann law. What is told in the Lindemann law? It is said that from one trophic level to another trophic level, only 10% of energy is being carried. So that is the basis for this question. Let us try to solve this part over here. They are saying that at the trophic level 1, How much energy is there? They are saying the question given is 100x kilocalories per meter square per year. How much will it get transported to the next trophic level? Only the 10% of it. So 10% of it is getting carried. What is the 10% of 100x? It is 10. So that means what? In the trophic level 2, how much energy? It is 10x kilocalories per meter square per year. This is in the trophic level. But they are asking in the third trophic level. Again in the third trophic level, how much energy is being transported? Yes, you are right. According to the 10% law, only 10% of energy is being transported. So we have to see what is the 10% of 10x kilocalories per meter square per year, and the answer is x kilocalories per meter square per year. So what is the right answer in this question? It is option 4. 4 is the right answer. I hope you all got this. How to solve this? It is just based on 10% law. Only 10% of energy is being carried to the higher trophic level. According to that, we can say that the answer is x kilocalories per meter square per year. And now, 149th question. This question is coming from Sexual Reproduction in Angiosperms chapter under the pollination. Yes. So I think here we are getting at least 2 questions are coming from pollination topic, if you observe. So this is the question from pollination and the diagram is for maize, alright, which is monoecious. And in maize, the pollination is solely done by wind, that is anemophily. Let us look at the options here. They are asking to mark the correct one. First option, cleistogamous. No, it's not cleistogamous, they are chasmogamous. Compact inflorescence showing complete autogamy. No, it is monoecious. As it is monoecious, gypnogamy is possible. They are unisexual. Autogamy, self-pollination is not at all observed in that. So not option 2 also. Option 3, wind pollination. That is what I told you in the beginning, right? Wind pollination or anemophily. So wind pollination plant inflorescence showing flowers with well-exposed stamens. And why are the stamens well exposed? So that they can easily release the pollen grains into their immediate environment, that is into the air, to carry out the wind pollination. So the answer is option 3. Coming to 150th question. This is the last question of botany. So match the following. Alright, so this question is coming from the morphology chapter. Rose, plum, peach, rose, all the 3 are showing perigynous flowers. Plum, peach, rose are showing perigynous flowers. P, okay, P is showing marginal placentation. P is showing marginal placentation, which means that the ovules are arranged over the margins. So P is marginal placentation for cotton. So cotton, it is belonging to Malvaceae family. So Malvaceae cotton is showing twisted estivation. It is showing twisted estivation. That means overlapping of sepals or petals is taking place in such a way that one margin is facing inside and the other margin is facing outside. That is the twisted estivation. Mango. Mango is the fruit coming under the drupe. Drupe is a true fruit. And drupe, it is produced from monocarpellary flower. And the characteristic feature of drupe is, in every drupe example, you can find that the endocarp is stony. that is a characteristic feature of drupe. Stony endocarp found in mango, also in coconut. And the edible part in mango is what? Yes, that is mesocarp. So D is 3, A is 2, B is 4, C is 1, D is 3. That means option 4. 4 is the right answer. Sorry, 3 is the right answer. So this is about the botany question paper. I hope you all found it easy and you have done very well, I hope. So this is about the botany question paper, and next we are going to move to the chemistry, but you can take 2 minutes of break and please stay tuned to the chat. Thank you.
Are you bored of your typical online classes that don't cater to your unique learning needs? Introducing our HighFlex program, online learning, offline doubt sessions. With HighFlex, convenience is on your side. With HighFlex, flexibility is on your side. With HighFlex, engaging classes are on your side. With HighFlex, accessibility is on your side. With HighFlex, stress-free learning is on your side. Step into the future of learning with our HighFlex program. What are you waiting for? Join HighFlex now and say hi to our experts in JEE, NEET and Foundation coaching.
నెంబర్లు చెప్పండి సార్, నేను ఇంకా అసలుకి ఫాస్ట్కి వెళ్ళి కానీ ఒక్కొక్క క్వశ్చన్ 60 మాత్రం డీటెయిల్గా చేసుకున్నాను. 60 మాత్రం డీటెయిల్గా చేసుకున్నాను, అది కొంచెం కష్టం. మిగతా అన్నీ
ఓకే
టిక్ 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 మీకే అది, ఎందుకంటే మీ దాంట్లోనే ఎక్కువ సాల్వింగ్ ఉన్నాయి. మీరే ఫార్ములా Hello, good evening to everybody. This is Raghavi, chemistry teacher from Knowledge Hub. So, how was the chemistry paper? As you all are saying, this is the game changer. It's not an easy one and at the same time consuming. So, let us get into the things fastly. Okay, 51 question number. In which of the following processes entropy increases? First one, a liquid evaporates to vapor. Temperature of crystalline solid lowered from 130 Kelvin to 0. That's not, as the temperature decreases, entropy decreases. So here solid is getting converted to 2 gases. So entropy increases here also, and in D also it increases. So the answer is A, C and D. That is first option. 52 question number. On heating, some solid substances changes from solid to vapor state without passing through liquid state. So they are talking about sublimation. The technique used for the purification of such solid substances based on the above principle is known as sublimation. This is from qualitative analysis. Next, identify the correct reagent that would bring about the following transformation. So here, when you take this one, double bond is getting converted to CHO. So first, when you take this one, when it is made to react with BH3 in presence of H2O2, then you will get anti-Markovnikov addition, CH2CH, double bond is getting changed to single bond and you will get OH. After that, when you take this one, if it is made to react with H2O2, PCC, then it gets converted to CHO. So I can say that here the answer is fourth option. First one, it is made to react with hydroboration followed by oxidation, and then partial oxidation. So fourth option, 54. The energy of an electron in the ground state for helium where n is equal to 1 is -6. Then that for an electron in n is equal to 2 state for beryllium ion in joules is. So we can say that E is equal to minus E1 into Z square by N square. Direct substitution, in the first case N value is 1 and in the second case N value is 2 and Z value in the first case is 2 and in the second case it is 4. Direct substitution will give you the answer as 3. Likewise, 55, this is match the following. Ethane, when you take, ethane is matched with 1 sigma bond. A should be matched with 1. So only one option A should be matched with 3. A is having 1 sigma bond. So I can say that the option is 1. A with 3, 1 and 2 options are there. But when you go for C directly, C should be matched with only 2. It has 2 pi bonds. So answer is first option. 56. For the reaction 2A giving B plus C, Kc is given. At a given time, the composition of the reaction mixture is given. Then, which of the following is correct? This is based on equilibrium. So, if I take here QC is equal to product concentration, that is B into C divided by A square. So, directly substituting, you will get QC value as 1. So, I can say that here QC is greater than Kc. So, first option. At the same time, 57, the E0 value for Mn plus 3, Mn plus 2 couple is more positive than that of Cr plus 3, Cr plus 2 or Fe plus 3, Fe plus 2 due to change of. Here in Mn plus 3, we do have D4 electronic configuration, whereas here we have D5 electronic configuration. So obviously the answer should be the first option. The E0 value for that is more because it is changing from D4 to D5. 58. Given below are 2 statements and they are asking the credibility of the statement. So the boiling point of the hydrides of group 16 follow the order. This order is correct. One of the bases of the molecular mass, H2O, is expected to have lower boiling point than the other members of the group. But due to the presence of extensive hydrogen bonding in water, it has higher boiling point. This is also correct one. So obviously the answer is third option. 59. Match the following once again. So isothermal process, when you take, it is at constant temperature. So A should be matched with 2. Only one option is there over there. So second option. Likewise, 60. This is a time consuming one. 1 gram of sodium hydroxide was treated with 25 ml of 0.75 HCl. The mass of sodium hydroxide left unreacted, they are asking. This is based on limiting factor concept. So here the number of equivalents of NaOH when you take, it is 1 by 40. At the same time when you take number of equivalents of HCl, it is equal to 25 into 0.75 into 10 to the power of minus 3. When you do it, it is 0.0182 you will get, and this is 0.025. So how much of NaOH is left out? Number of equivalents of NaOH left out is 0.025 minus 0.018. But this is not the answer. They are asking you the weight. So weight means number of equivalents 0.025 minus 0.0182 into 40. So that will give you the answer, and that comes to 250 grams. So in first session, this is the only question which is time consuming. Now when you take this one, again match the following. Here when you take the first one, it exhibits linkage isomerism. So A must be matched with 2. So the answer can be 2 or 3. Now if you see B when you take, it shows ionization isomerism. So it should be matched obviously with 3. So answer is 3. At the same time, among the group 16 elements, which one does not show -2 oxidation state? Here it's a direct NCERT based question. Polonium, because it's radioactive. 63. The compound that will undergo SN1 reaction. SN1 reactions are given when the carbocation is stable. So here this is a benzylic carbocation and also tertiary. So I can say that this is the answer. Likewise, 64. Which one of the following alcohol reacts instantaneously with glucose? So tertiary alcohols can do this work. So the answer is second option. 65. Match list 1 with list 2. Here 1 mole of H2O2 oxygen. We know that whenever we take 2 H2O to give 2 H2 plus O2, 4 moles of electrons are utilized. So for half, 2 moles of electrons, so 2 Faradays. So obviously A should be matched with 2. So we can say that A is matched with 2 in 2 of the options, 1 and 3. So out of 1 and 3 when you take, here if we go with B, 1 mole of MnO4 minus 2 Mn plus 2. So here 5 Faradays because change in oxidation state is 5. So directly the answer is third option. Now next one. The highest number of helium atoms is in. So 4 grams of helium means 1 mole. So N0. Here 2.27 means 0.1 moles. So 0.1 N0. Here 4 moles means 4 N0. And this is least because U indicates AMU. So I can say that they are asking highest. So answer is third option. Let us move to the next. Spin only magnetic moment is same for which of the following ions? So let us eliminate the things. B and C when you take, definitely they cannot be the answers because they do not have same number of unpaired electrons. We need to see number of unpaired electrons. When I take in B, Cr plus 2 as well in D, that is Fe plus 2, both are having 4 number of unpaired electrons. When number of unpaired electrons are same, magnetic moment will be same. So third option. 68. It is a direct one. Intramolecular hydrogen bonding is present in intra. Ortho isomers will have intra. 69. Failing solution AEs. This is a one which is a bit difficult because when we look at this we tend to tick the first one, but they are asking failing solution A, which is aqueous copper sulfate. Third option. Failing solution B is first, but they are asking A. 70. Match list 1 with list 2. So let us see first D. D is matched with size. So D should be matched with 2. D is matched with 2 only in one option, that is fourth option. Here this indicates magnetic quantum number. This indicates spin quantum number. So correct match is fourth option. So by looking at 1, you can eliminate the options. 71. Arrange the following elements in increasing order of first ionization enthalpy. These are the members of second period, but here there is an inversion. So finally the answer would be 4. Likewise, 72. This is also a direct one from physical chemistry. Which plot of ln K versus 1 by T is consistent with Arrhenius equation? So here we know that K is equal to A into e to the power of minus Ca by RT, or ln K is equal to ln A minus Ea by RT. So the slope of the graph should be negative. So the slope of the graph is negative only in this option. So second option is the correct one. 73. Arrange the following elements in order of electron negativity. So here when you take electron negativity, highest electron negativity should be there for fluorine. So answer should be in between these two. But we know very well that fluorine ke baad it is oxygen. So the answer is third option. Likewise, 74. Match list 1 with list 2. So here let us once again eliminate the thing. If I do D, our purpose will be solved. So when you take D, D should be matched with 3. So D is matched with 2 of the things like that, 1 and 2. So if I go with C, then the purpose will be solved. So C, when you take, OH is converted to ketone. So OH is converted to ketone with second. So obviously the answer is first option. So elimination. 75. Given below are two statements. Aniline does not undergo Friedel-Crafts alkylation. That's a correct statement because it forms a salt. Aniline cannot be prepared through Gabriel synthesis. That is also correct because it is an aromatic one. So both 1 and 2 are correct. So third option. So these are the first 25 questions in chemistry. The next 25 questions will be done with Sir.
Okay, thank you, ma'am. Hello everyone, this is Shravan Kumar, Senior Chemistry Faculty. Now I am continuing these questions. See, the 76th question is, in which of the following equilibria Kp and Kc are not equal? So I think all of you know this equation. Kp is equals to Kc into RT to the power of delta n. So not equal is given, that is a keyword. So not equal means delta n cannot be 0. So delta n cannot be 0 in which of the following? I think this is the one. Here it is a solid. For this, delta n is equals to number of moles of product minus number of moles of reactant. So number of moles of product is 2. Here number of moles of reactant is only 0 because of it is existed in the form of solid state. So that's why delta n is not equal to 0. So that's why the option for this one is third option. Now I am going to discuss about the next one. The most stable carbocation among the following is. I think all of you know this one. The tertiary carbocation is more stable than compared to secondary than compared to 1 degree. So that's why the out of all this is the 1 degree, this is the 3 degree, this is the 2 degree, this is also 2 degree. So the correct option is 2. Next one. See, the reagents with which glucose does not react to give the corresponding test or products are. So in this one, glucose, what the functional group is contained? That is aldehyde functional group is present. So in this aldehydes are not gives the reaction with sodium bisulfate along with Schiff's reagent. Tolerance is reduced, HCN is reacted, NH2OH is also reacted. So based on this, B and E is correct. So option 1 is the correct answer. Go for next. Given below are two statements are given. In this one, two complex compounds are given. In this one, first one is bounded with ammonia, second one is bounded with fluorine. But here ammonia is the strong field ligand, whereas fluorine is the weak field ligand. So, but even though here coordination numbers are 6, coordination number is 6, so that's why both are shows the octahedral complex. So first statement is correct. Next, second one, here it is a strong field ligand, all electrons are pairing is takes place, so that's why diamagnetic. But here it is a weak field ligand, so there is no pairing is takes place, so that's why it is a paramagnetic. So statement 1 is correct, statement 2 is also correct. So that means the correct option for this one is 3. Next, see this one. Activation energy of any chemical reaction can be calculated if one knows the value of. So that one is based on the given options, the correct one is temperature. I think based on this one only you are get the question, that is log K2 by K1 is equals to EA by 2.303 R into 1 by T1 minus 1 by T2. So this equation is total activation energy or rate constant is dependent on temperature. So that's why the second option is correct. Go for next. A compound with a molecular formula C6H14 has 2 tertiary carbons. So 2 tertiary carbons are present in 2,3 dimethyl butane. See, but means number of carbon atoms are 4. Here in this one, second position 1 carbon, third position is also 1 carbon is there. So this is the 3 degree carbon, this is also 3 degree carbon. So the option is 1. Next, 82. See, match the following. If you see the ammonia, it shows the pyramidal structure because of one lone pair of electron is present. So ammonia ion shows the trigonal pyramidal structure. Next, BRF5. Here in this one lone pair of electron is present, even though it is bounded with 5 fluorines. So the hybridization is sp3d2. So based on this one, the structure is square pyramidal. Next, xenon tetrafluoride. Here 2 lone pair of electrons are present, even though it shows the square planar. SF6 coordinate. Here it is the octahedral. So based on this, the correct option is 3. Next, the Henry's law constant values are given for ABC. Different values are given. The solubility of gases in water for the following order. But we know solubility product is inversely proportional to the KH. That is the Henry's constant is more, then solubility is less. So based on this, this is the least value for B, after this C, after this A. So the correct option is 4. See the next question. Which reaction is not a redox reaction? Redox reaction is nothing but if it is undergoes both oxidation and reduction reaction. That is known as the redox reaction. Here oxidation number 0, 0, but here hydrogen plus 1, oxygen minus 1. It is undergoes to the redox.
But if you see this one, barium plus 2, after the reaction is also barium plus 2. Chlorine minus 1, chlorine minus 1. Here sodium plus 1, sodium plus 1. Sulfate, entire ion. In this one, sulfur oxidation number 6, here also 6. No change in its oxidation number. So that's why the correct option for this one is 2. Next, 85th question. Given below are two statements are given. So in this one, the first statement, the boiling point of 3 isomeric pentanes follows the order. So what is the order? N-pentane. So all carbon atoms are present in straight chain manner. Next, isopentane. Isopentane. Third one, neopentane. So in this one, so the correct order is surface area is increases, then the boiling point values are increases. So first one is more, then second one, then compared to the third one. What is the reason? Because of branching is increases, the surface area is decreases. So that means statement 1 is correct, statement 2 is also correct. So the correct option for this one is 3. See the next question. The rate of a reaction quadruples when temperature changes from 27 to 57 degrees centigrade. Calculate the activation energy. Just now only we have discussed, that is log K2 by K1 is equals to EA by 2.303 R into 1 by T1 minus 1 by T2. Here K1 value is, if it is 1, then K2 value became 4 joule. Whereas temperature T1, 27 degrees centigrade, that is the 300 kelvins. Temperature T2, that is 330 kelvins. Because of 57 degrees centigrade is given, and R value also given. What is the R value? 8.314 into 10 power minus 3 kilojoules. Because of the answer is given in terms of kilojoules, so that's why I took that value. And log 4 value also given, that is a 0.6021. If you substitute all these values, then you get this one. Okay, next, see the next question. Consider the following reaction in a sealed vessel at equilibrium with concentration of N2 is given, O2 is given, NO is given. See the reaction. It is also lengthy question. See this one. So based on this, the equation is written as NO 1 by 2 N2 1 by 2 O2, or otherwise directly we can write here 2NO N2 plus O2. Here it is a 2 moles, 0, 0 at initially. At equilibrium 2 minus alpha, alpha, alpha. So this is a 2 minus 2 alpha. So commonly 2 and cross multiplied, then we get 1 minus alpha, alpha by 2, alpha by 2. Then apply the equilibrium constant K, alpha by 2 into alpha by 2 into 1 minus alpha. So Kc, that is alpha by 2 into alpha by 2. Alpha by 2 is common. And here I forget to take this value, power 2 into 1 by 1 minus alpha whole square is equals to. If you substitute these values, then we get 1.6. Under root of this, alpha by 1 minus alpha is equals to, we get 1.6. Here alpha by 1 minus alpha is equals to, here 2 is also there, that is 1.26 we are getting. 2 is cross multiplied, alpha by 1 minus alpha is equals to 12, 5, 2. Cross multiplication, while solving the alpha, then we get the value is 0.717. So by solving this one, you get the exact value. Next, see, during the preparation of Morse salt solution, Morse salt is nothing but ferrous ammonium sulfate, FeSO4, NH4 taken twice, SO4 dot XH2. This is the ferrous ammonium salt. Which of the following acid is added to prevent hydrolysis of Fe plus 2? That means if you want to add the water, then it undergoes to the hydrolysis, then we get Fe plus 2 is converted to Fe plus 3. But in this one, hydrolysis is stopped by the addition of dilute sulfuric acid because of common ion effect. When you add the sulfuric acid, due to common ion effect, the backward reaction takes place. So backward reaction is nothing but hydrolysis reaction is stopped. That means the correct option for this one is dilute sulfuric acid. See the next one. A compound contains 32% of A, 20% of B and remaining percentage of carbon. Then the empirical formula of X, that is here element A, B, C is given. Percentage of A 32%, B 20%, the leftover quantity is 48. Then empirical formula, first step, the percentages are divided with their atomic weight. For A 64, for B 40, for C is given 32. Cancel with twice, 16 3s are 16 2s are. We get 1 by 2, 1 by 2, 3 by 2. So when you solving this, we get 1 1 3. That means number of A units are 1, B units are 1, C units are 3. So the correct option is 4. Next, the pair of lanthanide ions which are diamagnetic. Already according to NCERT, direct question, the answer is this one, third option. See the next one, major products A and B formed in the following reaction sequences are. So first of all, reaction with PBr3. So when it is taken, alcohol is OH is replaced with, CH3. Here what we get here? Br. After this, when it is undergoes to the alcoholic KOH, dehydration reaction takes place. Then we get this one is the product. So the correct option is based on this one. Third option is correct. Okay. So this is the option. This is a product A and this is a product B. Next. The plot of osmotic pressure versus concentration is given and slope is also given. What is the slope value here given? 25.73 liter bar per mole. Now what is the value we have to calculate? Temperature we have to calculate. So if you take the osmotic pressure, pi is equals to CRT. So this is Y is equals to X into M. So M is nothing but slope. So M, in place of M we can substitute the RT. RT is equals to 25.73. T is equals to 25.73 by R. So already R value we know that is given 0.083. If you substituted this, then you get the value in terms of degrees. You get the value in terms of Kelvin, but converted into degrees, then you get 37 degree centigrade. Now see the question, 93. Given below are certain cations using inorganic qualitative analysis, arrange them in increasing group number from 0 to 6. So according to practical chemistry, direct question, the answer is this one. Okay. So direct question from the practical chemistry. So the next question is, the products A and B obtained in the follow reactions respectively. So in this one, if alcohol ROH PCl3, same question just now only we have discussed, OH is replaced with Cl plus H3PO3 is the bright product. This is the product A. Next, same alcohol reacting with PCl5, then we get RCl plus POCl3 is the byproduct along with HCl. So the product A and B are in this one. So the correct answer is H3PO3 and POCl3 is the correct option for this question. See the next one. For the given reaction, what is the major product? So here the given component is strong oxidizing agent. So the role of strong oxidizing agent is when the double bond is present, that is broken, and this hydrogen is also oxidized with KMnO4. So that means we get the product directly,
That's the fourth option. Because of double bond is broken and hydrogen is also get oxidized, then we get carboxylic acid. Next, the work done during reversible isothermal expansion of 1 mole, number of moles is given 1 mole of hydrogen gas at temperature is given 25 degrees, that is a 298 kelvins. Initial pressure is given P1, P2. P2 is given 20 atmospheres. P1 is given as 10. Direct formula, expansion W is equals to minus 2.303 nRT log P1 by P2. If you substitute, n value already is there, 1, temperature 298 Kelvin, P2 20, P1 10. If you substitute all these values, then we get the answer is fourth one. Direct substitution, there is no change. See, next, identify the major product C formed in the following reaction sequence is. So, in first reaction, iodine is replaced with cyanide. So, what's the answer? CH3, CH2, CH2, Cl plus NaCl, NaI is formed. So, this is the A. A is undergoes to the partial hydrolysis, not complete hydrolysis. By complete hydrolysis, we get the carboxylic acid. But here partial hydrolysis, then we get CH3, CH2, CH2, CONH2. Amide is formed due to partial hydrolysis. Here it is reacting with NaOH followed by bromine. That is Hoffman bromide degradation reaction. So rearrangement reaction is takes place. Then we get the product that is propyl amine. So CH3, CH2, CH2, NH2. This is a product. So here this is a named reaction, that is Hoffman bromide degradation reaction. Next one. Identify the correct answer. That is, out of all this we have to choose the correct answer only. This is also statement wide of question, type of question. So out of all this, second option is correct, because of canonical structures are shown by the carbonate ion is only 3. But dipole moment of NF3 is not greater than that of here NH3. So here printing mistake is there, sorry. So the first one is incorrect statement. Third one is also incorrect statement. Because of ozone shows only 2 canonical structure, but here is given 3. BF3 has non-zero dipole moment. It is also wrong statement. So the correct option is 2. See the next one. Mass in grams of copper deposited by passing of 9.6487 amperes current through a voltmeter containing copper sulfate solution for 100 seconds. So according to Faraday first law, the weight of substance is formed at respective electrode is directly proportional to quantity of current is passed. So based on that one, we get this one. C is nothing but current in ampere, that is a 9.6487 ampere. Time is given in 100 seconds. Here W is equals to electrochemical equivalent, that is equivalent weight 63 is given by 2, n factor is 2 from copper sulfate, by Faraday is given 96,487 into C value 9.6487 is given, time is given 100. By solving this, 2 zeros, sorry. So we get, by solving of all this, then we get 0.315. If you want to solve this one is, we get 31.75. It is cancelled with 1000 times, 10 times. So we get, I think I only make the mistake. So that's why the correct answer we are getting 0.315. Okay. So by 100 we get 0.3175. Okay, this is the answer. Next one. Given below 2 statements are given. The first one is here, hexaminocobalt is given homoleptic. Homoleptic means if same type of ligands are bounded with central metal atom, that is a homoleptic. Whereas here 2 type of ligands are present, different type of ligands are present, that is known as the heteroleptic. So first statement is correct. See the statement 2. Only one type of ligands are present, that is known as the homoleptic. Different type of ligands are present, that is known as a heteroleptic. So statement 1 is correct and statement 2 is also correct. So based on this one, the third option is correct. So this is the chemistry paper. Now we are going to discuss, let's take the 2 minutes break. Continuation part will be taken by the zoology faculty. Okay, thank you.
NEET Elite, your gateway to success in medical entrance exams. Are you an ambitious 10th, 11th grade student dreaming of a seat in top medical colleges, including AIMS? Join our NEET Elite batch. Look no further. Our comprehensive coaching program is tailored just for you. 1 year and 2 years integrated hybrid program. NEET Elite ensures cutting-edge curriculum, expert guidance, and personalized attention. But that's not all. We understand the importance of a comfortable learning environment with a focused and nurturing atmosphere. Enjoy the convenience of residential and transport facility. Limited seats. Enroll now to secure your seat in NEET Elite at Knowledge Hub by Sri Chaitanya.
Are you bored of your typical online classes that don't cater to your unique learning needs? Introducing our HighFlix program, Online Learning.
Hello everyone, welcome back to the session. As you all know, I am Dr. Deepak, Senior Zoology Faculty here at Knowledge Hub. So I'll be taking you through the zoology part of today's NEET 2024 question paper. So let's start with the first question. So here's the first question, match the following. You can see there is list 1 and list 2. So there is Pleurobrachia, which is matched with the Ctenophora. And then there is Radula, which is matched with Mollusca. Stomachord is matched with Hemichordates, and air bladder is matched with the Osteichthyes. Right? So you all know Radula is a file-like rasping organ present in the mouth of mollusks, and it's a direct question from animal kingdom. So the option is 4. Right? Moving to the next question, again match the following. So if you observe the question paper, in the zoology part, basically, we have around 20 questions which are given in the form of match the following, and around 8 questions are statement-based questions. All the questions are from NCERT, but few questions are little lengthy, but overall the paper was easy and it is not very lengthy. It definitely takes time to go through the questions, but it's not so lengthy. Yes. Next question, match the following again. So list 1, list 2, this is a question from reproduction. Non-reproductive IUD. Non-reproductive IUD will be Lippes loop here. Then copper releasing IUD. Copper releasing IUD examples are copper T, copper 7, and you have multi-load, and hormone releasing IUD and implants. So what is the hormone releasing IUD over here? What is the hormone releasing IUD over here? We have hormone releasing IUD, which is LNG20, and implant is progesterone. So what is the correct option? Option 2 is the correct. Moving to the next question, again match the following. This is the third match the following here. So list 1, list 2. So you have alpha-1 antitrypsin. So alpha-1 antitrypsin is for emphysema. Next we have Cry1AB. So Cry1AB is for, is it for cotton bollworm or is it for corn borer worm? It is for your cotton bollworm. Next Cry1AC is for corn borer worm, and enzyme replacement therapy is for ADA deficiency. So what is your correct answer, students? What is the correct answer? So option 1 is the correct answer. Next, match the following, list 1 and list 2. This is the fourth match the following in the question paper. So common cold is for rhinoviruses. Hemozoin granules are found in the plasmodium. Next, viral test for typhoid and allergy dust mites. This is a question from Human Health and Disease. So what's the correct option over here? So option 1 is correct. Alright. Next, match the following. So you can see all the questions till now which we have been discussing are match the following. So the first one, cocaine. So cocaine, what is the correct match in the list? Cocaine is for erythroxylum coca. Right, it is obtained from erythroxylum coca. Next, heroin. What is heroin? for in the options given over here? What is heroin for the, in the question you can see heroin is for papaver, somniferum, then morphine is for effective sedative in surgery, and marijuana is from cannabis sativa. This is also a question from human health and disease. So what is the correct option? Option 2 is the correct answer. Next, again match the following, right? So list 1 and list 2 over here. This is a question from cell cycle. This is a question from cell cycle and cell division. So here you can see there are different phases of prophase 1, right? There are different phases of prophase 1 given in the question. So we have a mnemonic over here, Li-za-pa-di-dia, right? So leptotene, zygotene, pachytene, diplotene and diakinesis. So according to this, you are supposed to match the specific characters which are seen. So here the correct option is option 1, because if you see diakinesis is for completion of terminalization of chiasmata, and then we have pachytene for appearance of recombination nodules, and we have zygotene for synaptonemal complex formation, and we have leptotene for chromosomes which look like thin threads. Next, question number 157. This is a statement based question. Till now, whatever the questions we have discussed are match the following. Now this is a statement based question, and this is from reproduction. So you can read the two statements which are given over here. Statement 1, the presence or absence of hymen is not a reliable indicator of virginity. True or false? This is a true statement. Next, statement 2, the hymen is shown during the first coitus only. This is a false statement, right? Because hymen can be torn due to various reasons. So statement 1 and statement 2, so what is the correct option over here? Option 1. Statement 1 is true, but 2 is false. Next, question number 158. What is the correct product of DNA dependent RNA polymerase to the given template? As we all know, mRNA is always complementary to the template strand, right? mRNA is complementary to the template strand, except for T, right? Because in place of thymine, it is always replaced with uracil. So here the correct option is option 3. Yes. Again, there is a match the following from human physiology. So here you are supposed to mark lung volumes and lung capacities with the respective formula. So here we have the correct option is option 3 here, but how it is option 3? If you see the expiratory capacity, right? Expiratory capacity is equal to, expiratory capacity is the tidal volume plus expiratory reserve volume, right? Then what is inspiratory capacity? Inspiratory capacity is tidal volume plus inspiratory reserve volume. So you can match expiratory capacity with tidal volume plus expiratory reserve volume, and then we have inspiratory capacity which you can match with the tidal volume plus inspiratory reserve volume, right? Next, what are the two other things which are left here? Functional residual capacity and vital capacity. So functional residual capacity is expiratory reserve volume plus your residual volume, and we have vital capacity which is equal to, vital capacity is equal to inspiratory reserve volume plus expiratory reserve volume plus tidal volume, right? So it goes with option 1. So what is the correct answer over here? Option 3. Next, which of the following are autoimmune diseases, right? So over here you have 5 options. First one is myasthenia gravis, which is an autoimmune condition, right? It is an autoimmune condition. Next we have rheumatoid arthritis, which is also an autoimmune condition. Gout, it is not an autoimmune condition because gout is caused due to the accumulation of uric acid crystals. Next, the fourth one is muscular dystrophy. Muscular dystrophy is also not an autoimmune disease because muscular dystrophy is a genetic condition, it's a genetic disorder. Next, the last one is systemic lupus erythematosus, SLE, which is also a autoimmune condition which is affecting the multiple systems of human body. So how many of these are autoimmune? A, B and E. So what is the correct option? Option 4 is the correct. Next question. So here we have statements. If you see question number 161, first option, see the 4 statements over here. Annelids are true coelomates. True or false? Annelids are true coelomates. Yes, they are true coelomates. Poriferans are pseudocoelomates. False. Poriferans are not pseudocoelomates. Right? Poriferans are acoelomates. Ascalmenthes are acoelomates. This is also false because ascalmenthes nematodes are pseudocoelomates. Platyhelminthes are pseudocoelomates. Platyhelminthes are not pseudocoelomates. Right? So this is also false. So choose the correct option. So what is the correct option? Option 1 is correct. So it goes with option number 4. So students, I hope I am not very fast. Right? Let's move on to the next question. This is also again a statement based question. Question number 162. So statement 1 and statement 2. Read the statement 1. In the nephron, the descending limb of loop of Henle is impermeable to water and permeable to electrolytes. Is this the truth statement? No. It is the false statement. Why is it false? Because it should be vice versa. Right? Instead of descending limb of loop of Henle, it should be ascending limb of loop of Henle. Right? So this is the false statement. Next, if you see the statement number 2, the proximal convoluted tubule is lined by simple columnar brush bordered epithelium and increases the surface area for reabsorption. So here it should be cuboidal epithelium. This is not columnar epithelium. Right? So this is also a false statement. Right? So both the statements are false. So option 4. Right? Next question. Again, there's a match the following. Right? So list 1, list 2, you have to match. These are the scientific names again given from animal kingdom. So here the correct answer would be option 4, but how? We have Pterophyllum. Pterophyllum is the angelfish. Myxine, what is myxine? Myxine is hagfish. Pristis is the sawfish. Exocetus is the flying fish. Right? So the correct answer is option number 4. Moving to the next question. Which of the following is not a steroid hormone? Direct question, which was asked on human physiology, chemical coordination. So over here there are 4 options. We have progesterone, glucagon, cortisol and testosterone. Right? So which of the following is not a steroid hormone? We know progesterone is a steroid hormone. Right? It's a steroid hormone. And cortisol and testosterone are also steroid hormones, but glucagon is not a steroid hormone because glucagon is a peptide hormone. Glucagon is a peptide hormone, so option 2 is correct. Moving to the next question, again there's a match the following, right? So let's match. So here you have in list 1, there are 4 enzymes. And list 2, you can see different types of bonds. So lipase is an enzyme which is going to break. Lipase is an enzyme which is going to break peptide bond, ester bond, glycosidic bond, phosphodiester bond. Come on. Lipase is going to break ester bond. Nuclease will be breaking which bond? Nuclease will be breaking phosphodiester bond. Protease, peptide bond. Amylase, glycosidic bond. So the correct answer would be option number 1, right? Again match the following. So here you are supposed to match with list 1 to list 2, and over here they have given different diseases, different conditions, different syndromes, and here you can see the related chromosomes. Alright. So first is your Down syndrome. Down syndrome is basically caused due to trisomy 21. Right? Presence of one extra 21st chromosome, right? So here I am going to match this Down syndrome with 21st chromosome. Then comes thalassemia. So alpha thalassemia and beta thalassemia are 2 different types of thalassemia. Basically thalassemia is a autosomal recessive condition. So alpha thalassemia and beta thalassemia. Alpha thalassemia is caused due to the deficiency or the mutation of the gene which is present on the 16th chromosome, and we have beta thalassemia which is caused due to the deficiency or mutation of the gene which is present on the 11th chromosome. Now what is left here? Klinefelter syndrome. Klinefelter syndrome is due to the presence of one extra X chromosome, right? So the ploidy goes like this, XXY, there is presence of one extra X chromosome in the males. So Klinefelter male will be showing female characters, right? So what would be the correct option? Option number 1, right? Yes. Next question, please. This is again a direct question from biotechnology. The TI plasmid of Agrobacterium tumefaciens stands for, very direct question, very easy question, right? It's no point of getting confused here. It's option number 1, tumor inducing plasmid. Next. This is a question again from reproduction, a statement based question. You have assertion and reason. So just read the assertion over here. Breastfeeding during initial period of infant growth is recommended by doctors for bringing a healthy baby. True or false? Yes, this is true. Next. Reason. Colostrum contains several antibodies absolutely essential to develop the resistance for the newborn baby. This is also true. And is it a right explanation or wrong explanation? It is the right explanation. So both A and B are correct, and R is the correct explanation of A. Option 3 over here is correct. Moving to the next question. A direct question from reproduction again. Which of the following is not a component of fallopian tube? So basically, what are the different components of fallopian tube? You have fimbriae. Fimbriae are finger-like structures which help in collecting the ovum. Then we have a funnel-like structure which is called infundibulum, which leads to ampulla and isthmus, right? So you can just see here which of the following is not. Obviously, uterine fundus is not the part of fallopian tube, right? And the remaining 3 are the parts of fallopian tube. Next, 170. In both the sexes of cockroach, a pair of jointed filamentous structures called anal cerci are present on. So anal cerci are basically present on the 10th segment, right? They help in sensation, right? They are kind of sensory structures present in the cockroach, and these are not same as anal styles. Anal styles are basically seen only in the male cockroach, right? But anal cerci are found in both male as well as in the female cockroach. Next, which one of the following factors will not affect the Hardy-Weinberg equilibrium? So it's again a direct question, which is not going to affect. So gene migration will be affecting, genetic recombination will be affecting, genetic drift also will be affecting. It's a direct question again, option 2, right? Constant gene pool will not affect the Hardy-Weinberg equilibrium. Question from evolution. Next. This is a question from brain, from your neural control and coordination, right? Again, there's a match the following. You have to match with list 1 to list 2. So different parts of brain. So you have pons, hypothalamus, medulla, and the cerebellum. So you're matching pons with connecting different regions of the brain. And then we have hypothalamus, which is the neuroendocrine organ. It has neurosecretory cells. And then comes medulla. So medulla can be matched with the centers because it has different centers which controls respiration, heart rate and different other activities. So controls respiration, gastric secretions. Next, we have cerebellum, which is going to provide additional space for neurons. Also, it regulates the posture and balance. So what is the correct option? Option number 4 over here is correct. Next question. The flippers of the penguins and dolphins are the example of. Yes. This is also a question again from evolution topic. So there are 4 options given over here: convergent evolution, divergent evolution, adaptive radiation, natural selection. So what is the correct answer over here? So the flippers of penguin and dolphin, they are basically used for swimming, right? So the flippers of penguin and dolphin are used for swimming, right? So these structures are functionally same but structurally different. So the option would be option number 1, that is convergent evolution. Because these structures, are they homologous or analogous? These are analogous structures. Next, this is also again a very easy question from human heart. So following are the stages of pathway for conduction of an action potential through the heart. So here you can see AV bundle, Purkinje fibers, AV node, bundle branch, SA node. So it is going to start with the SA node, sinoatrial node, which is called pacemaker of the heart, and then it goes to AV node, and from AV node it is going to AV bundle. From AV bundle it is going to reach the bundle branches.
Are you bored of your typical online?
Bundle branches, which are also called His bundles, and from finally bundle branches to the Purkinje fibers. Purkinje fibers. So what is the correct option over here? Option number 3. Moving on to the next question. Yes. The following diagram showing restriction sites in the E. coli cloning vector PBR322. This is a question taken from biotechnology, which is picked up from NCERT, and it's given over here. So what is the role of X and controlling the copy number? So it is called ORI, right? Origin of replication. And then the Y part or the Y gene, it is called ROP part. You will find these letters directly in your NCERT textbook, right? So option number 4 here is correct, because the gene XORI is responsible for controlling the copy number, which is linked to the DNA, and Y is ROP, which is nothing but it's regulating the process of it is regulating the the multiplication or replication of plasmid. All right. Moving on to the next question. This is also
Bored of your typical online classes that don't cater to your unique learning needs? Introducing our HighFlix program, online learning, offline doubt sessions. With HighFlix, convenience is on your side. With HighFlix, flexibility is on your side. With HighFlix, engaging classes are on your side. With HighFlix, accessibility is on your side. With HighFlix, stress-free learning is on your side. Step into the future of learning with our HighFlix program. What are you waiting for? Join HighFlix. Are you bored of your typical online classes that don't cater to your unique learning needs? Introducing our HighFlix program, online learning, offline doubt sessions. With HighFlix, convenience is on your side. With HighFlix, flexibility is on your side. With HighFlix, engaging classes are on your side. With HighFlix, bored of your typical online classes that don't cater to your unique learning needs? Introducing our HighFlix program, online learning, offline doubt sessions. With HighFlix, convenience is on your side. With HighFlix, flexibility is on your side. With HighFlix, engaging classes are on your side. With HighFlix, accessibility is on your side. With HighFlix, stress-free learning is on your side. your typical online classes that don't cater to your unique learning needs? Introducing our HighFlix program, online learning, offline doubt sessions. With HighFlix, convenience is on your side. With HighFlix,
सेटिंग ही चेंज नहीं करेंगे.
are you bored of your typical online classes that don't cater to your unique learning needs? Introducing our HighFlix program, online learning, offline doubt sessions. With HighFlix, convenience is on your side. With HighFlix, flexibility is on your side. With HighFlix, engaging classes are on your side. With HighFlix, accessibility is on your side. With HighFlix, stress-free learning is on your side. Step into the future of learning with our HighFlix program. What are you waiting for? Join Are you bored of your typical online classes that don't cater to your unique learning needs? Introducing our HighFlix program, online learning, offline doubt sessions. With HighFlix, convenience is on your side. With HighFlix, flexibility. Are you bored of your typical online classes that don't cater to your unique learning needs? Introducing our HighFlix program, online learning, offline doubt sessions. With HighFlix, convenience is on your side. With HighFlix, flexibility is on your side. With HighFlix, engaging classes are on your side. With HighFlix, accessibility is on your side. With HighFlix, stress-free learning is on your side. Step into the future of learning with our HighFlix program. What are you Are you bored of your typical online classes that don't cater to your unique learning needs? Introducing our HighFlix program, online learning, offline doubt sessions. With HighFlix, convenience is on your side. With HighFlix, flexibility is on your side. With HighFlix, are you bored of your typical online classes that don't cater to your unique learning needs? Introducing our HighFlix program, online learning, offline doubt sessions. With HighFlix, convenience is on your side. With HighFlix, flexibility is on your side.